Okay, so now that we've looked at different components that might be in the membrane, we have to return back and think about why the membrane, how the membrane is formed. And so what we look at when we're talking about cells is we're typically looking at what's called a lipid bilayer. And so that's different from what you might expect, for instance, for a normal, for a standard amphiphilic molecule, such as the ones we talked about earlier in the semester when we were talking about the hydrophobic effect and how micelle formation occurs. And so part of this is, this depends on the structure of the amphiphilic molecule and the structure of the amphiphilic molecule and the relative widths of the nonpolar tail and the polar head group. So if we think of an amphiphilic molecule such as this, where we have, again, we have this nonpolar tail group here, and then we have this polar head group here, so we'll call this polar head group, and then nonpolar tail, okay? All right, so if we look at this, and this is a little exaggerated, but it also kind of makes a good point. So if we look at this, the polar head group is going to have a larger width than the tail. So what you have is you have this cone that looks kind of like this. Another thing you can also think about this is, we'll call this a phospholipid slice of pizza, okay? So if we think about how these, something with such a structure, such as this pizza slice, would form the closest packing so as to exclude as much water, you can think very easily of, again, a whole pizza, right? Where each slice is a phospholipid, right? And the cone around the phospholipid. Okay? And so what you see then basically is how you can get sort of the structure of this micelle, which is shown on this slide on the right-hand side. So the close packing of the hydrophobic tails excludes water. Again, the entire idea of micelle formation comes from the hydrophobic effect, in which this is not an enthalpic effect. There's very little enthalpic contribution between the interaction of these hydrophobic tails, but it's an entropic effect because these hydrophobic nonpolar tails are now avoiding interaction with the water. So go back, if you need a refresher on that, go back to the videos that discuss the hydrophobic effect, because this is really important, right? Because the point is, we want this closest packing between the tails, such as shown here in the second, in this figure B, this panel B in the slide here. Because if we expand out, outwards, you then form cavities that water can now fill. And again, that defeats the purpose of keeping water out of interaction of the nonpolar tails to avoid this unfavorable entropic effect. We can also have this sort of more of a elongated structure, an oval structure, but that also creates its own gaps where water can fill in, okay? By contrast, glycerophospholipids have more of a rectangular shape where because there are several tails attached to the glycerol, this takes up more space, and if we can think of adding another tail to this polar head group, now it's more even, and we can think of this as a more rectangular shape, such as this, and then instead we end up with in our pizza analogy. So now we have a sheet pizza. We have our polar head group. And our nonpolar tails that point towards each other and interact with each other. And so if you extend this again to micelle formation by these glycerophospholipids, you can see sort of this oval shape, but if you extend the oval shape, you can extend this all the way around until they connect. Right? So again, if within this there are two polar head groups, so one here, and then the tails point towards each other, okay, and this encompasses all the way around this sort of donut shape. And then if we close this, then what we have is a lipid bilayer. Like this. Right? So just continue this all the way around the circle. And what you see is you have a lipid bilayer. So now you have a membrane which can separate the environment, the surrounding environment, from the inside. Okay? This is exactly what the membrane does for the cell. It allows for enclosure, so definition of the boundary of the cell and enclosure, so that different cell processes can actually change the environment inside the cell, and it has very little effect depending on the surroundings. So we can have different environment here, which may be important for different physiological processes. And so from this, we have a structure like this. It's called a liposome. Okay? Where this is just a membrane, where this is just a lipid bilayer that's encapsulated itself and formed an inside and an outside. And so this is a liposome, and we can think of that. So additions to this liposome, such as the addition of DNA inside, or proteins, or gradients, concentration gradients, such as proton gradients across this membrane, or other things that are important for physiological processes, we can see, we can think of this as sort of the primordial beginning of a cell. Once we start adding functionality to the inside of this and using this for benefit for biological processes, then that's sort of the primordial beginnings of a cell. Okay?